afternoon, everyone. Welcome to today's live practice session for the course Fundamentals of Bioprocess Engineering. So before we move on with the session, I'll just wait for a couple of minutes to. So if you have any doubts from previous lecture videos or from previous session discussion, you can just post in the chat box. And I hope the audio is clear. If there is some interruptions, you can let me know. Okay, so once again, welcome you all to the second live practice session for the course Fundamentals of Bioprocess Engineering. So for those of newly joined this week, my name is Guru Priya Ramesh and I'm a Prime Minister's Research Fellow in the Department of Civil Engineering, IIT Madras. So I have my BTech and MTech in biotechnology, and currently I'm pursuing my PhD in the field of anaerobic digestion research. So with that, the course content for the week 2 was on material and mass balance, stoichiometry of the bioprocess, where there were information on how to quantify substrate consumption, product formation, as well as biomass growth, and there was also lectures on metabolics and energy balance. So if you have any specific doubts from any of week 1 or week 2 contents, you can just post in the chat box at any point in the meeting. So with this, I told that we'll be having a question answer based discussion session. So today we have some problems to solve, but before that we are just going to start with very easy and introductory theoretical questions, like choose the best answer or fill in the blanks kind of questions. We'll just see, just to get used to the topic, and then we'll move on to solving problems. Okay, as I told, the first question is, in a batch reactor, which statement is the most correct? You can just post the option in the chat box like you did the last week. batch reactor, accumulation is not always zero because it doesn't operate under steady state conditions. And B also means the same. In a batch reactor, there is no continuous input and continuous output. We just feed it, let the reaction get over, and then only take out the product. So there will be accumulation, and ideally there is no case of input and output generation in a batch system. And talking about the mass balance is applicable to all biochemical processes. So it always applies whether it's a batch, wet batch, or a continuous system. It doesn't matter. So C is the right answer. Okay, so the next important concept that we use in many of the problems is the degree of reduction. So one question about that. Correct again. Degree of reduction. A higher degree of reduction means the compound is more reduced. So, do you have an idea of why we talk about degree of reduction? What is the significance of it? We'll also have a few more questions related to it in the later part, but just to start with. So first of all, we all know what is degree of reduction. Degree of reduction means the equivalence of available electrons in a material per gram of carbon atom. Right, that is what we define degree of reduction to be. But why is it important? Okay. Briefly, degree of reduction tells us how much of energy can be derived from a particular substrate or a compound. So that's an indicator of how much energy we can derive. And having said that, as I told, we'll have a little detailed discussion on this degree of reduction, electrons, energy content, how all this is related in the later part of this class. Okay, one more question just to sum up all the concepts. You can just give like 1A, 2B, 3C, 4D, like that, whatever matches with what in the chat box. exactly. So COD, the chemical or the biochemical oxygen demand, uh, generally tells about how much of oxygen is needed for complete oxidation. So it gives us an estimate of the required oxygen that we have to be supplying to the system. And biomass yield coefficient is y x by s, which tells us about how much of biomass is produced per gram of substrate consumed. So this helps us to see where the substrate consumption is directed in the biochemical mechanisms. So if the more substrate is consumed but biomass production is not that much, then it means the energy is going, the consumed mass is going somewhere else. So in order to close the balance, we need to find out where the mass is going. So that is an indicative of how efficiently the consumed substrate is converted into biomass. Similarly, YPY is talks about how much of the consumed substrate is converted to a product. And P by O, it refers to the number of ATP formed per atom of oxygen consumed. In other terms, we can also tell that as number of ADP, that is phosphorylated, per atom of oxygen consumed. Both are the same because ADP is phosphorylated to become ATP. And degree of reduction, as we saw in the previous slide, is the available electron content per carbon atom. Okay, so next question. Two cultures have identical biomass yield on glucose. The culture A consumes twice as much oxygen. What can you infer from this statement? guesses? What would you comment on? Let's say we have culture A and culture B. What would you comment on? Okay. Okay, so here we have three answers. One is oxygen in the culture is getting consumed in some other metabolic process, which is partially okay. Uh, the next is culture A needs more oxygen to produce the same yield, but B doesn't need. It means maybe B is more efficient in product formation. But here, I hope that the product that you mean here is the biomass, because apart from that, we are not talking about any product being formed here. And finally, culture A less sensitive for glucose production because it uses more oxygen for energy production. But here we talk about glucose consumption. They have yield on glucose, means per gram of glucose consumed they produce the same amount of biomass. Culture A and B produces same amount of biomass per gram of glucose consumed. But in order to do that, culture A requires twice as much as oxygen as what B uses. So there can be multiple reasons for it. I just wanted to see what are all you are able to think of. The main thing is culture A has lower maintenance efficiency, which means the maintenance demand is more. So more of the oxygen that it consumes go towards the survival of the cell than towards the biomass generation or the growth. Similarly, there can be some byproduct formation towards which the oxygen consumption is rerouted and not towards the biomass. And similarly, maybe the uh, P by O that we talked about. P by O refers to number of ATP produced per atom of oxygen consumed. So that could be low for culture A. So it needs more oxygen to produce the same amount of ATP or to have the same amount of energy. So either it's about the maintenance efficiency or rerouting towards some other byproduct formation, or the fact that culture A has a lower P by O ratio. Also there can be some kind of a metabolic deflection, like there can be some leak in the electron transfer chain, or some other unwanted cycles may be in action, to which ATP is generated but it is going towards some unwanted cycles. These all can also be the reason. So here when I have mentioned explicitly maintenance energy, because that's a very common thing that we can think of, and also it depends on the conditions in which we are going to have the bacteria. Maybe in the conditions that we culture they might be identical, but culture B can be more adaptive towards the environment than A is. So in that case A will require a higher maintenance energy. That is what I have mentioned here as lower maintenance efficiency. So don't confuse between the maintenance efficiency and maintenance energy demand. When the maintenance energy demand is high, it somehow means that the maintenance efficiency is very low. Okay, I hope it was clear. If there are doubts, please let me know so that I can repeat. So in the meantime, next question. A fermentation shows decreasing oxygen uptake, constant glucose consumption, and increasing ethanol formation. What does this indicate? This is comparatively easier than the previous one. Right. So, this indicates a shift towards fermentative metabolism, like from aerobic to anaerobic. That is right. And similarly, it is kind of an indicative to the Crabtree effect. We can assume that there might be excess of glucose So to consume which, let's say if there is yeast, it takes the fermentative route for um, consuming the overflow of glucose. On the other hand, there might be oxygen limitation in the system. So when there is oxygen limitation, the uptake also decreases, but glucose is constantly consumed because the metabolism shifts towards fermentation, not aerobic respiration. Aerobic respiration, though it is energetically very rewarding, but because oxygen is limited, there can be a shift. So that's the right answer. Okay, those are few questions about some basics and theory. So from here we will just see some problems. Similarly, I'll give you some time to solve the problems as well, and then we can discuss the answer. So a batch bioreactor initially contains 500 liter of medium with 20 gram per liter substrate. After fermentation, 12 gram per liter substrate remains and 4 gram per liter of biomass has been formed. Now here we have to assume that there are no byproducts and there is no evaporation. And you have to find out the amount of substrate consumed, the biomass yield if 4 gram per liter biomass was formed from the consumed substrate. This is a simple question. You can just have a diagram with input and output. No need of any equations for this question. So I'll give you 5 minutes time before we can discuss the answer. Okay, exactly. So initially we have around 20 gram per liter substrate concentration, and the total reactor volume is 500 liters. So we have 10,000 gram of substrate. Similarly, after the fermentation, the concentration is 12 gram per liter, but the reactor volume is going to remain the same. It's 500 liters. So it is 6,000 grams substrate still left, which means there is 4,000 grams of OKG that is consumed. So the amount consumed is 4,000 and the amount formed is 2000. How? Because in a 500 liter medium, 4 gram per liter concentration is biomass, which is 2000 grams. So 2000 grams of biomass is produced when 4000 grams of substrate is consumed, which gives us a biomass yield as 0.5 gram per gram. Okay, next question is about, first was about mass balance. So here we talk about degree of reduction. Find the degree of reduction of glucose and acetic acid, and you can give your inferences about glucose and acetic acid based on their degree of reduction values. Hope you have the formula for glucose. Glucose is C6H12O6 and acetic acid is CH3COOH. just compare the degree of reduction of both the compounds and give your inference. Okay, so this is the formula for finding out the degree of reduction. W is the number of carbon, X is hydrogen, Y is oxygen, and C we don't have in both acetate and glucose, and W here is the um, carbon equivalent. So based on that, both acetate and glucose have degree of reduction to be 4. So now I'll wait for another few minutes. I hope you got right until this. You can just tell me like what you infer from both glucose and acetate having the same degree of reduction. one of the previous questions we saw what is degree of reduction. So based on that, do you have any opinion on what does it signify? Okay, so theoretically it means that they would yield the same amount of energy, that is theoretically, yeah, we can predict that, and the available electron for exchange is same, the oxidation potential is also same. Yeah, that also theoretically we can infer, but practically the bonding in glucose and acetate is different, right? So when we talk about the functional groups, we have OH groups, we have an aromatic ring uh, and stuff in glucose, whereas when we talk about acetate, it is going to be a carboxylic group and a CH3COOH it is, so it has a carboxylic group. When we talk about the internal energy distribution, it is depending on the different functional groups and bonds that are present in each of the uh, chemical compound, and also this only decides their shape as well. So breaking both is not practically the same thing to do. So that's why both glucose and acetate can show similarities, but they also have a great amount of metabolic differences. So when we talk about glucose, it comes into the picture in glycolysis, but when we talk about acetate, it only comes during the TCA cycle. So practically and theoretically speaking, there are differences because of all these factors. Just by comparing two substances or degree of reduction of two substrates, you cannot tell that they are going to behave identically the same. So here each carbon atom carries the same number of available electrons. They have the same theoretical reducing power, but metabolically they are not equivalent. So that you have to keep in mind whenever you compare substrates, or whenever you plan your reactions for any substrate of this matter, you have to have a clear understanding of what is a theoretical prediction and how it can vary in your actual bioprocess system. Only to introduce this concept, I had this degree of reduction question. I hope it is clear. So next concept. There are any doubts, post it in the chat box. I can answer them in the meantime. So next is another question. If you just have a, let's say this is a biomass. Voice is breaking. Is it clear now, or do others also face this issue? Okay, for the previous question, the thing I was telling was, theoretically both glucose and acetate may seem to behave the same in a bioprocess system, but their entry points in the metabolism, or how the organisms can metabolize them, depends on their chemical structure, chemical bonding. So based on that only, the actual energy that you can derive from the compound is going to be, and that will be slightly vary. So there will be a slightly upper hand of glucose than acetate, and similarly when we talk about glucose, the chain starts from glycolysis, whereas when we talk about acetic acid, it comes only in the TCA cycle, like it has to be converted to the acetyl CoA, and then only it can integrate into the metabolics to produce ATP. So based on this, theoretically we will not get exactly the same amount of energy from both the compounds, though practically you will not get the same amount of energy from both the compounds, though theoretically they appear to be similar. This concept you have to have in mind whenever you are going to design a process for yourself. Whatever you see in theory may not exactly be reproducible in the lab as well. It's just a guiding factor, but you also have to have a look on other factors that will affect your bioprocess. You cannot use acetate in place of glucose and say you are going to get the same results, same amount of product, or same amount of biomass yield. So in that aspect you have to be clear. So that was about the previous question. Now estimate the COD for a biomass with the given formula: C5H7O2N. 0.42 gram per gram is the right answer. And this formula we directly have, like how to find a COD, but I hope you all know from where we derive this formula. So when we have any component with a molecular formula of CN, HAOB and NC, this formula is applicable as it is. Like here the N is the number of carbon, A is the number of hydrogen, B is the number of oxygen and C is the number of nitrogen in the given molecular formula. So from where we derive this is, let's say we talk about complete oxidation of a compound which has a molecular formula CnHaOb and Nc, then there is some amount of oxygen required for the complete oxidation. So in the reactant side you will have the substance to be oxidized and oxygen, and when there is complete oxidation the product is going to be CO2, carbon dioxide and ammonia and water. So these three are going to be the products of complete oxidation. Okay, when we talk about the chemical oxygen demand, we are going to estimate the total amount of oxygen that is required to completely oxidize the given substance. So in that case, assuming complete oxidation, your reactants are going to be the substrate for which you have to find the COD and oxygen, and the product is going to be carbon dioxide, ammonia and water. Okay, now when you are going to draw a equation balance, stoichiometric balance for this equation, you will find out what is the coefficient of oxygen going to be, because for carbon dioxide and ammonia it's pretty easy, because in your reactant side you have carbon only in the biomass for which you are going to find the COD. So the amount of carbon, which is the n number of carbon, has to be present as CO2, so you will put NCO2. When you talk about ammonia, you have C number of nitrogens in the reactants, so the stoichiometric coefficient of ammonia is going to be C in that case. And then when you talk about H2O, you have oxygen in both carbon dioxide and water, and you have your hydrogen in both ammonia and water. So here you can just write the terms relatively, right? So when you do the essential balancing, you will see that you will have A minus 3C by 2 number of hydrogen. Similar thing when you are going to do a stoichiometric balancing for the amount of oxygen in the reactant side, you will get a term, and that term is essentially in the numerator of the COD formula. Okay, so this is that term, that 2n plus 0.5a minus 1.5c minus b by 2 is going to be the coefficient of oxygen in the complete stoichiometric equation that you are going to write for oxidation of any biomass. And then what is COD? COD is the gram of or the mass of oxygen required. So you are multiplying it with the molecular weight of oxygen and dividing it with the molecular weight or mass of the biomass itself. So that brings you to this formula given here, which is COD. You need not mug up this formula entirely, just understand the concept that for if there is complete oxidation, you are going to have the biomass and oxygen in the reactants and the other components in carbon dioxide and nitrogen and water in the products. Those are going to be the only products of your complete oxidation. So when you talk about COD, it is gram oxygen per gram biomass. In that case, you are just multiplying and dividing it with the appropriate molecular weights. And then finally, for the given biomass formula, the COD requirement is 1.42 grams oxygen per gram biomass. Is that clear, or any doubts? Okay, if there are any doubts, you can post in the chat box anytime. The next question is, for glucose consumption, if 5 mole oxygen is consumed, how many moles of ATP are formed by oxidative phosphorylation? Are you able to try? If not, you can just let me know. I will discuss the answer. Or anyways, I'll just wait for a couple of more minutes to see if you can answer it. Okay, so for this question, uh, so classically we have the ratio that the P by O of glucose is 3. So based on that, the 5 moles of oxygen is again consumed, so which we can have it as 2b, like when we have an equation we can have it as 2b, and the number of ATP formed is going to be 2b into p by o. So 3 into 5 is going to give you 15 moles of ATP. The next question is, a fed batch reactor initially contains 1000 liters of medium with 10 gram per liter substrate. During the run, 500 liter feed containing 80 gram per liter substrate is added. At harvest, the reactor contains 1500 liters with 12 gram per liter substrate. So the biomass found is 8 gram per liter in the final broth. The initial biomass was negligible. You have to find the substrate consumed, the biomass produced, the biomass yield, and whether if there is any substrate that is unaccounted, or is everything perfectly balanced. This is also mass balance based question only.
Boleh.
Has anyone found the substrate consumed?
Right down.
Okay. Okay, so the initial substrate is 1000 liter medium with 10 gram per liter, so it's 10,000 grams. And again, the what was fed is 500 liters with a concentration of 80 gram per liter, so 40,000 grams was then added. So now you have 50,000. From that, 18,000 was remaining, so 32,000 grams has been consumed, or 32 kg has been consumed. So when talking about the biomass produced, we have 8 gram per liter biomass, and finally the volume is 1500 liters. So 8 gram per liter into 1500 gives us 12,000 grams or 12 kilograms, and the yield is going to be 12,000 grams is produced totally from 32,000 grams, or 12 kilogram from 32 kilograms. So it is 0.375 gram per gram is the biomass yield. Unaccounted substrate is, we had 32,000 initially, but only 12,000 is converted it to biomass. Similarly, there is a remaining 22,000 grams or 20 kilograms which is unaccounted. So this unaccounted can be used for maintenance, or for some other. I haven't told the entire story here, so there might be production of some other product here. Apart from the biomass, we can have some kind of alcohol acid production. There can be maintenance, or simply it can be converted into CO2 itself. So you have unaccounted substrate, and it can be going to any of the streams. Okay, the next question is, there is a substrate with the formula C4H8O2, and it is converted to a product with the formula C3H6NO3. I want you to estimate the theoretical maximum product yield. So the product is C3H6O3 and the substrate is C4H8O2. So from this, you have to estimate the theoretical maximum product yield, and to make it easy, you denote the theoretical maximum product yield as Fmax. So from the given data, compute the Fmax value. Okay, so here Fmax is not the exact denotation. It depends on however you have your stoichiometry assumed. So for to solve this question, you have to have the stoichiometry of product formation. You have to have your substrate, which is you have a carbon or energy source, a nitrogen source, and an oxygen. That gives you biomass, product, carbon dioxide, and water. That is the typical equation. So from that equation, if you have to find the product yield, what you do is you, like we did in the previous, you have to find the oxygen demand. So in that case, what you did, you took the coefficient of oxygen, multiplied it with the molecular weight, and divided it with the molecular weight of the biomass itself. So in this case, similarly, you have an equation when you have to find the product yield, which means the amount of product formed per substrate consumed. So in that case, you have to take the coefficient of um, your product, multiply it with the molecular weight of the product divided by the molecular weight of your substrate, assuming that your substrate doesn't have any coefficient. If it has, it's going to be the coefficient of your product into molecular weight of your product divided by the coefficient of your substrate into molecular weight of the substrate. This is what we ideally assume by using a stoichiometric equation. Okay, so here when you talk about electron balance, your degree of reduction will come into picture. Okay, uh, here you have some electron balance, and in general cases you assume your nitrogen source to be ammonia. So in that case you will have a electron balance. So from the electron balance you can derive equations for product yield and biomass yield. Okay, so the equation that I am talking about is what your uh, substrate plus oxygen plus your nitrogen source is going to give you your biomass, product, carbon dioxide and water. So this is what you will ideally get. For this equation you will ideally assume your nitrogen source to be ammonia, and you will have a electron balance equation. So where will the electron balance equation come from? When you talk about your substrate, you will have degree of reduction of your substrate into whatever is the coefficient of carbon here. Okay, in whatever we assumed as per the lecture equation, I am just giving those denotations, but whatever denotations you take, it's completely fine. Similarly, minus, if A is the coefficient of oxygen here, it's going to be minus 4A, and here you will have your coefficient of your biomass into degree of reduction of your biomass and your coefficients of your products into degree of reduction of your products. So here you will have some coefficient, and here you will have some coefficient. So for your product you will have some, let's say your product is C X H Y O Z and some N. So here, apart from the coefficient term, you will also multiply this X. Okay, you are exactly following whatever is given in the lecture, then the coefficient of your product is going to be F. So that is what this star will be replaced by F. I want you to find what is the maximum value of F that is possible, which is F max. So when is your F max is going to come into the picture? You assume whatever you put in is going to be converted into product. That means this term will become 0. So for the maximum product yield, F becomes F max and other terms becomes 0. Okay, so from that relation, so what is that relation essentially? This is the relation. F max is equal to W into degree of reduction of your substrate divided by J into degree of reduction of your product. So here the degree of reduction of your substrate you can find out with the given formula. Similarly for your product, and you will also know the number of carbon atoms. It is 4 in the substrate and 3 in product. So the degree of reduction of your substrate is 5 and for product it is 4. So when you are going to multiply and find do the multiplication and division, the F max value is going to be 1.67. So even when you don't mug up exactly what is the formula for theoretical maximum product yield, you should know to frame an equation. You should know that there is going to be a substrate, a nitrogen source and oxygen, from which you will get a biomass, product, carbon dioxide and water. This is like you have to have it very standardly in your mind. Based on whatever the question is, you will have to add or eliminate terms. When they say that the substrate is completely oriented towards biomass, then you assume that there is no product formation form. Like when we discussed for the COD estimation problem, in COD estimation there is going to be no product, there is going to be complete oxidation. So in that case you should know which terms to have and which terms to not have. And similarly, whenever the nitrogen source is not specified, you are going to assume the nitrogen source is going to be ammonia. I hope this answer for this question is clear. So I will just go through the other few questions and upload these slides so that you can practice them, because I think we will be finishing soon. So a microorganism grows aerobically on glucose with ammonia as nitrogen source. See, in this question we have explicitly mentioned that ammonia is the nitrogen source, and the molecular formula for the biomass is C4.8 H7.2 O2 and N. Okay, experimental analysis shows that 65% of the glucose carbon is converted into biomass and the rest goes to carbon dioxide. So here you have to determine the stoichiometric equation of the reaction. So ideally what you will have here, you have your substrate. So you have C6H12O6 plus ammonia plus oxygen. This is what is going to react. Okay, because they have given that it glows aerobically on glucose with ammonia. So you have glucose, air and ammonia. And the product they have mentioned, they have only given about the biomass, so you need not worry about any other product that is formed here. Your duty is to write about this formula like C4.8 H7.2 O2.0 and N0.8. Okay, and there is DCO2 there is CO2 formation and water formation, that you know. So what you do here is write down the complete equation and write balances for each of the equation. You can assume any stoichiometric coefficients. So you can assume any number of stoichiometric coefficients. So here we have given the coefficient for oxygen is A, for ammonia it is B, for your biomass it is C, for carbon dioxide is D, and for H2O it is E. Now your duty is to determine what these coefficients values are. So for that you have to have your carbon balance. So carbon balance is there is and you are assuming that 6 like 1 mole of glucose is going to react. Okay, so that's why you don't have a coefficient here. So when you do a carbon balance, in reactant side there is 6 carbon, whereas in the product you have 4.8 into C plus D. That is one equation. Okay, and it is given here that 65% of the glucose carbon is converted into biomass, which means out of this 6, 65% goes to the biomass. So 6 into 65 percentage is the 4.8 into C. That is what it is mentioned here. 4.8 into C is 65% of this 6, which is 3.9. So with that you can find what you have found out what is C. Similarly, you know that C here, you just substitute the C value here and find out what is D. So now you know what is C and what is D from carbon balance. Next you go to the nitrogen balance. So nitrogen balance, nitrogen is present in ammonia and in the biomass. In biomass it is 0.8 into C, you know the C value, here it is B. So B is equal to 0.8 C, and then with that you find out how much of ammonia, how much moles of ammonia is used. Similarly for hydrogen balance, you have 12 hydrogen here and then 3B number of hydrogen here, which gives you 7.2 C number of hydrogens plus 2E. Here except E you know all other values, so you can substitute all other values from the previous step and then you will finally have your E value. And then when you write an oxygen balance, you will figure out what your A is. In some cases they might have given you the value of your respiratory quotient, so your D by A is going to be respiratory quotient. If that is given, then you can, when you have directly found out D, you can just use that relation to find out your A either ways. So to solve any stoichiometric equation, the first step is to write out a balance for each of the element that is present. This is one question. So the next question is, in a batch experiment, 50 gram per liter biomass is formed in 10 hours from 100 gram per liter glucose consumed. Okay, so initially it has 100 gram per liter substrate, the biomass formed is 50 gram per liter, and the time duration is 10 hours. If biomass yield on glucose without maintenance is 0.6 gram per gram, how much glucose was diverted to maintenance and non-growth functions? It is similar to some of the questions that we saw before. Glucose required for biomass is 50 divided by 0.6, which is for 50 gram of biomass with the yield of 0.6 gram per gram, you would have needed 83.33 grams of glucose, but 100 grams was actually consumed, so the remaining 16 grams should have been gone to the maintenance or non-growth related functions. This is relatively a simple question. The next question is, a substrate which is glucose is converted into a biomass with this given formula, and the product for 1 mole of glucose, assume that 0.5 moles of biomass and 0.8 moles of product are formed. You have to calculate the oxygen coefficient. So this is the equation. You have glucose reacting with oxygen and ammonia. Here you are not given what the nitrogen source is, so you have to assume that it is ammonia. So here you don't know the formula for your biomass, but you know for your product, so it's up to you how you denote them. So I have denoted them as zero point 0.5 into x, which is the biomass, 0.8 into p, which is the product, because I am assuming that for 1 mole I am taking only 1 mole of glucose here, so it should only give me zero point 0.5 moles of biomass and zero point 0.8 moles of product. And for the unknowns, I have just given assignments of A, B, D and E. So to find the oxygen coefficient, which is A, you again go with your balancing steps. You balance each of the element and finally you can find out what is A. So these were the questions that were prepared for today. I hope they were useful to you. So if there are any doubts, you can still ask. So were the questions useful for in today's class? Were you able to solve? Was it too easy or too tough? If you can give me some feedback, I would be able to prepare accordingly for the next session. If there are no more doubts, thank you for joining. You can leave the session. Uh, Pia, which equation are you exactly talking about? The heat reaction formula when oxygen is electron acceptor. Thank you, Rajeshri. Glad you liked it. Okay, yeah, I have a few. I have not talked about any of the energy balances in this. So this session was completely on stoichiometry and mass balance. So the next session, when we start with, we will start with this energy balance as well. So because of time constraint, I have planned only questions from these topics for this session, and I think we completely stopped it. We had exact time to only discuss those questions. So the next time when we come, the first 2 or 3 questions that we solve will be based on this concept. That is how I have planned. Is it okay? Yeah, okay. Because it was already too much of a long content, we needed enough time to solve the problem. So that's why I just saved it for the first few minutes of next week's session. We'll surely see this concept in detail. Any other doubts? Okay. If anyone has any other doubts, you can ask, and the PPT of this session will be uploaded, as well as the video of the session will also be uploaded, which I think you can access. So if there are no more doubts, thank you for joining in for the session. Hope to see you in the next session as well. If there are any doubts or any suggestions about the session as well, you are free to post in the chat box anytime during the meeting. So yes, thank you for joining.